Hello, everyone, and welcome to today's live broadcast, Smart Plastics, Improving qPCR Reliability and Reproducibility. My name is Christy Jewell. I'm with LabRoots, and I will be moderating today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Eppendorf. For more information on our sponsor, please visit and click on the Eppendorf logo on the left side of your screen. Now, before I begin, I would like to encourage you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Additionally, we have a few polls for you, and we welcome your participation. If you have any trouble seeing or hearing the presentation, just click on that support tab found at the top right of your presentation window, or just use that Ask a Question box and let us know you're experiencing a problem. Today's presentation is educational, and it does offer continuing education credits. Following the presentation, those credits can be obtained by clicking on the tab at the top right of your presentation window and follow the process to obtain your credits. Before I pass on this presentation to our speakers, I would like to point out that you can read full biographies on Dr. Tim Schommartz, Dr. Michael Pfaffl, and Dr. Rafal Grzeskowiak. Gentlemen, the stage is yours.
Thank you very much for this nice introduction, Christy. I am Tim, and as introduced, I would like to send out a warm welcome from Eppendorf for this educational webinar. We are very happy that you all took the time to join us today for about 60 minutes so that we can give you some insights on how to improve your qPCR reliability and the influence of consumables on qPCR workflows. We are extremely honored that we are joined today by Professor Michael Pfaffl from the Technical University of Munich. So we will start with an introduction to the MIQE guidelines, the relevance for improving qPCR reproducibility, and some examples. And from that, we will then continue to take a deeper look into the influence of consumables on qPCR workflows, some tips and tricks, what to look out for when choosing consumables, and how to handle with them. But first, I would like to get started with Professor Pfaffl. So I'm handing over to Professor Pfaffl. The stage is yours.
Thank you very much for this nice introduction, and thank you very much out in the world. I really appreciate it to take place in this webinar. And what I want to talk for the next, let's say, around about 10 to 15 minutes is about the MIQE guidelines, a short introduction, the relevance, and a few examples. And let's start. And the big question is, what is already on the market? Are there quality control strategies and standardization initiatives in genomics, transcriptomics, or molecular testing, or even beyond? And there are some examples here. The first one was probably the MIME, the standardization of hybridization microarrays. Then we had the MIQE and the digital MIQE guidelines. They're dealing with real-time PCR, RT-qPCR, and of course digital PCR. And there are a lot of consortia dealing with quality control in NGS, in next-generation sequencing. And there are also initiatives from the OECD or WHO about testing. We, as our group here in Germany, are involved in the MIQE guidelines and also in standardization NGS, so how to get valid biomarker signatures from liquid biopsies and how to standardize NGS. And we published in this area quite a few publications. So we have these quality problems and reliability problems as well today in SARS testing, and I want to show you some comments which are going around. This is some comments from the European Union, where the Commissar complained about that we have been working hard to analyze information on the quality assurance of the coronavirus testing and devices that are currently being used. The results show that there is a mismatch between the existing quality and what could be expected to ensure the good performance of the tests. So we have a huge problem, and also the scientists complaining that there might be false negative results, 1 out of 5 samples during corona testing. This is going quite broad through the press in UK, where the scientists raised concern about quality control, and hence we got a position paper from Stephen Bustin and Tanya Nolan, also courses of the MIQE guidelines, about RT-qPCR testing, and they have a big concern about misinterpreting the results of corona testing. So what is the big question? What is the central problem in qPCR? It's undefined variability. You know it from your results as well. If you have various experimental group or treatments, often you have single groups with high error bars or huge variability, and the question is where this variability is coming from. And hence I try to group these problem areas in diagnostic PCR in 3 parts. So we have the pre-PCR part, the PCR and the post-PCR, and all of these groups more or less impact the variability and the reproducibility in PCR. So pre-PCR, of course, a huge impact of sampling technique, sample quality, the extraction quality and extraction efficiency of your nucleic acid. Then, of course, how to prove it. You have to be sure that the RNA and the DNA quality, quantity, and integrity, and also purity of your sample is of high integrity, and also the experimental design is very important. Jumping over to PCR, of course, protocol, cycling profile, meaning fast cycling, rapid cycling, has a huge impact and often produces unspecific products or even primer dimers. And now we are coming of more or less to the topic of the day, how the consumables and hardware are impacting tube plates, tips, sealing, robotics, and so on. They have a huge impact on point 7, the assay validation, meaning how specific is your assay, how sensitive do I come down to a sensitivity of a few dozens of molecules, how reproducible is my assay in comparison to other labs, to other researchers, or in a worldwide ring testing, and of course what is this limit of detection and quantification. And also from point 8 to 13, is more or less the impact of post-PCR, meaning how can I increase my reliability after I perform PCR, how I handle the data, how I handle outliers, the normalization strategy, and so on, efficiency and statistics at the very end. But today we are focusing on this point, consumables and hardware, and how this reacts on assay validation and reproducibility. So hence, a bunch of researchers were sitting together long ago. This was, I think we started 2007 to 2008 to get collect all these problems. And these are 12 researchers from, I can say, all over the world. Most of them are coming from Europe, from Czech Republic, England, Belgium, Sweden, me from Germany, and of course Stephen Bustin, first author, and also Carl Wittwer, he's the inventor of the light cycler system. And we were sitting together, we were discussing what do we need? Of course, we need guidelines. Guidelines, the so-called MIQE guidelines, the minimum information for publication of quantitative real-time PCR experiments. But what does it mean? What does MIQE stands for? And this is a set of guidelines that describes the minimum information necessary for evaluating qPCR experiments, and the much deeper and broader goals are to provide guidelines for authors, reviewers, and editors to measure really the technical quality of the submitted manuscript or the paper or the work you've done, to establish a clear framework to conduct qPCR experiments. It should support experimental transparency, increase reproducibility in your lab, but also between labs, even worldwide, and it should promote consistency, more comparable and more reliable results. And during these days, also to standardize the nomenclature. And to bring it to the point, the MIQE guidelines should increase the reliability of qPCR results and should ensure the integrity of scientific work, of course, with major focus on the biological relevance. And if you see here on this slide, it worked. It worked out. Up to now, we have, these are very data from the last weekend, we have now more than 9,800 citations of this paper. It means all these citations are coming from the big, let's say, academia, countries like United States, Germany, Europe, United Kingdom, China, and from all over the world. You see here a lot of countries are using the MIQE guidelines. So let's come to the content of the MIQE guidelines. So they are divided in 9 major headings. We call it the MIQE checklist. So these headings are experimental designs. Here you see them, or you don't see them, but I depicted it in larger letters. So experimental design, sampling, nucleic acid extraction, reverse transcription, target information, the primers, the protocol, assay validation, and at the end, the data analysis and the statistics. And normally, we got information about the PCR efficiency, and we also got a lot of information in the papers about the normalization strategies, about the software use, but the real PCR protocol and the consumables used in the assays are often forgotten. So hence I want to focus today on the MIQE checklist, on number 7, on the PCR protocol, and here I want to focus here what is the impact on the plates, tubes, even is the reaction setup from a robot or is it manual? I would just want to point out these 2 points. And long ago, 2008, we published a paper about these, and this was the effect of plate position and plate type and sealing system on real-time PCR. This is a paper from 2008. It's already 12 years old, but during these days, we were working with the master cycler from Eppendorf. This was the days where Eppendorf was producing a real-time PCR machine. So we were also had one of these machines, and we want to test it, the impact of white plates versus transparent plates. So we had 8 strips with white and with transparent wells. Here you see we set up, or we assembled kind of a virtual plate in a 96 setup with transparent and white sections, and then we performed, in all of these we performed standard curves. Here we have transparent white, and you see depending on the white or the transparent plate itself, we received very nice standard curves. All have a high efficiency with 93, 94 percent of PCR efficiency. The regression was good, the slope was good. But if we go deeper in the data, we saw a huge difference between some samples here and some samples there. And if we divide this, we see that these samples are coming from transparent plate, and these more or less as the amplification curve is coming from white plates. All of these dilutions here from left to right were always the same concentrations from hundreds, thousands, up to millions of copy. And you clearly see here the standard curves, the reproducibility is good. But what I want to point out is the information output from these white plates. And if you look here on the data, we call this during these days the amplification efficiency in terms of increase per cycle. And if we have here the so-called transparent, we have here hundreds or in the hundreds, and in the white plates we have in the thousands, meaning we have a huge significant increase in the amplification efficiency in the readout of the fluorescence information. During these days we used the cybergreen assays. The gene doesn't matter, but this was very reproducible. And if you summarize this data, you see here the white and the transparent plate that the white plate receive a much earlier crossing point of more than one cycle earlier. I have to say, these were all performed in one block at the same time with the same reactions and so on. And also, the standard error here in the white plate is slightly smaller than in the transparent plate. And what we further analyzed here is the difference between heat sealing and adhesive sealing. You know these differences. And again, the heat sealed samples received lower errors. So meaning you really have an advantage using good consumables, using white plates, using heat sealing. These results show it very clearly. And at the last slide, we also, during these days, we were thinking about to buy a robot. Okay, it was during these days, it was very expensive, and therefore we made experiments where we tested our hand pipetting. And this is a plate which we analyzed with a software of a former Ph.D. student. For me, it's RT-qPCR. But what you see here is more or less the differences in hand pipetting over a 96 well plate. We were not using 8-channel pipette, we were single-channel pipette. All is the same concentration. And what you here see is the size of the rings. Either it's underestimated, it's the red color, overestimated, it's the green color. And what you see here is the X's is more or less the significant from all the other significant differences from all the other results. And you clearly see that you have a significant outlier in these border areas. We all know this, that we have these border effects on plates, independent of any assay worldwide. But this should a little bit depict the significance of maybe using a robot, okay? So this is just to keep you thinking about reproducibility in a plate setup. Okay. Let's come to the end. This was just an introduction about the importance of MIQE guidelines, and hence I want to point out of our MIQE-qPCR iBook, and I have to say that since this week we have the 4th edition online since Monday this week. You can download it. This is a free book. We have it now on the market since about 5 years. We have 20 chapters in this book, and the title is How to Apply the MIQE Guidelines, a visual and interactive practical qPCR guide. You can download it for free. We have a lot of chapters in, and of course one chapter is about qPCR consumables, and this was more or less written by the Eppendorf team, by the co-authors and co-speakers as well from today, from Rafal and from Muriel. And with this slide, I'm going to hand over to my co-speakers. Thank you for your attention and have fun during this webinar.
Yeah, thank you, Michael, for this very interesting insight into the MIQE guidelines as well as the experimental data on how consumables can influence the outcome of qPCR experiments. So I will take over from here, and in my first slide, I would like to take a step back, because when I thinking back at my times working in the lab and doing assays of some sort, it might have been qPCR, but it could technically be anything else. And when the experiment was not working, you go back and you do a lot of troubleshooting. And I was thinking about a lot of things, right? So first of all, you think about yourself, you think about the sample handling, the preparation, then you question maybe the samples itself. So are these quality samples? Maybe something went wrong with the samples while processing it. Then another big factor is reagents. So are the reagents still okay? Have they been frozen correctly? And of course, you are also thinking about the lab equipment. If you do any measurements, is the pipette calibrated correctly, stuff like that. But another important influence on assays that is often overlooked is the actual consumables, because technically there is no assay which does not get in contact with consumables of any sort. Most of those consumables are made out of plastics. Of course, there are some made out of glass, but most of them are made out of plastics. And during this webinar, we will take a closer look at that plastic does not equal plastic and how they can influence the outcome, especially of qPCR results. So, as I said, consumables are in technically all the workflows, and the reason what makes them very influential is that they are obviously always in direct contact with the sample or with the reaction mix itself, and by such can directly influence vital parameters and consequently the performance of all your experiments. So a high quality consumable assures high quality and also reproducible results, obviously. So if I talk about consumables, which is like a very broad term, what consumables am I referring to? So, and if I break it down to classical qPCR workflow, we could say that, okay, the classical consumables that I think all of you are using in your workflows are some sort of tips for the pipetting. No matter if you do it manually or automated, you will always use some sort of tips. Then you are using single tubes of different sizes depicted here. Some of you will likely use tube strips. I think most of you are using some sort of plates. And I think that also a lot of you listening are using seals. So this is a overview of the most common formats. If we talk format, I will take a little excursus here in terms of nomenclature, not necessarily only important for qPCR, but in general, because when I came to Eppendorf and when people were talking to me about consumables, sometimes I was lost with the, let's say, vocabulary. So I want to, especially for the plates, I want to give you a little bit of a deeper insight here, because plates are usually divided into several types of plates. Of course, there are PCR plates. They can have different formats, usually 96 well, sometimes 384 well. Then there are plates which take a larger volume. These are those, like, let's call it bigger plates. Those are called deep well plates, or in short, DVP. And then we have so-called microtiter plates, or MTP plates, sometimes also called just a microplate. So those are plates which can take a lower volume than deep well plates, but which are classically also not PCR plates. So for deep well plates and microtiter plates, there are different so-called well shapes, and you might have seen that already. So there is the so-called flat bottom. This is for most of the ELISA plates which are in use, they have a flat bottom. Then we have a round bottom and so-called V-shape. And depending on the application, it might make sense to use a plate with the correct bottom. The PCR plates usually do not come with a different bottom shape, so they have a adjusted V shape, so to speak. They always have this PCR plate shape. So that was a little excurse, just that you know what I'm talking about. If I say DVP or deep well plate, that we are on the same line here. So, but as I said, so what I did before is I was breaking down the consumables related to their format. But that might not be the best way to look at the performance criteria for consumables if you look at a certain workflow. So the better way is to break down the consumables and to sort them to the actual workflow steps. So if we look at a qPCR workflow, there are consumables which are holding your samples and reagents. These are mostly single tubes of a larger format, so starting from 1.5 ml tube usually up to maybe even 15 ml tubes if you have a lot of reagent, and then there are plates. These can be PCR plates, microtiter plates, or deep well plates. But what is in common is that these are vessels which are mainly used for storage and often also long-term storage of reagents and samples in a freezer or in any environment. And then on the other side, there are the plates and vessels which are mostly only used for a short time, so for the assay itself. And those are single tubes, strips, and plates, which of different formats. And what happens in those things is that the actual setup of the assay has been done in those vessels, as well as the readout. And since the readout is involved there as well, I put seals there as well because there is, especially for plates, there is a need to close them if they go into a qPCR cycler. And the connecting factor is the tips. So with the tips, you are connecting the vessels which are holding your samples and reagents with the vessels where you do your actual assay. So, and by the nature of these different tasks, there are different prerequisites for those consumables. So consumables which are holding samples and reagents, especially for long-term storage, one of the most important things is that they ensure sample integrity. So you don't want your precious sample to be lost during the storage process. Then, of course, there is the sample safety in terms of processing. I think all of you know, for example, if we talk about single tubes, if you want to spin down a sample, this tube is better be centrifugation stable. I remember a wonderful episode from my study times in a test lab where a whole batch of tubes more or less exploded in a centrifuge filled with blood. It was big fun to clean that up. And you want to also ensure in those storage-related vessels that you have maximum sample or reagent recovery. So you don't want to lose any of your precious sample or reagent to the vessel itself. User safety is important, especially if dealing with hazardous material. And of course, you want to have a good handling. Ideally, you want to have a good handling with one hand, and you also want to have a good tracking of your samples if we talk about plates. Okay, if we look at the tips, I think first and foremost thing is precision. So if you want to do a qPCR setup, and if we talk manually, but also in a automated fashion, of course you want to pipette the correct volumes. So the first thing that you want your tip to be is ensure maximum precision and accuracy when it comes to pipetting. Then, of course, it's sample safety. So cross-contamination is, I guess, the biggest enemy of every qPCR experiment. And you want to have also sample and reagent recovery. And as I will not do a deep dive into tips, two things that I want to say here is, of course, it is extremely important, the combination between tip and pipette. So if you have tip and pipette from the same manufacturer, usually you have the best system, which ensures maximum precision, and in terms of cross-contamination, I was always a big fan of using filter tips for all my qPCR experiments. All right, and if we talk about the final assay itself, about the assay vessels, of course you want to have optimum assay results, which means you want to have optical brilliance for the readout, but you also want to have very good heat transfer so that the cycling works very well for your qPCR experiments, as well as easy and safe handling during the assay setup. And of course, usually a qPCR experiment is not done in a vacuum, so it usually doesn't end with a single qPCR experiment. We all know, working in the lab, that there is more than one experiment needed. There are subsequent experiments needed, experiment lines. So ideally, you want all of those consumables to have the same quality, the same performance for each and every experiment. So one common thing you want to have for all of those things is that you have an optimized, let's say, and very reliable batch-to-batch consistency. So if you get one batch of plates, you want them to perform exactly the same like the one you used a month before. Okay. So and since the qPCR plate is, I think, the thing that what I see is mostly used during qPCR experiment. What I want to do is I want to describe, in my view, the perfect qPCR plate. Of course, this differs depending on the experimental setup, but ideally, the plate has a solid polycarbonate frame, which with an easy to read engraving, so that you can see, okay, what is my position D7 without taking ages to find the column and the row. Your plate, if we talk about handling, should have a NZ/ format. That is a standard thing. If you ask yourself why all of those plates, 6-well plates, 24-well plates, 48-well plates, they all have the same format, is that they have the so-called NZ/ format. If you have plates in this format, this makes them compatible with most of the systems out there, with most of the instruments, and it's also very automation friendly, which leads to the barcode. So more and more stuff is currently getting barcoded, especially if we talk about high throughput. So ideally, you want to have a barcode option for a plate. So if we talk about the well, what is very important there is that you want to have a well which is very thin-walled, so which ensures optimal heat transfer, so that the cycling is fast and precise. And Professor Pfaffl already introduced the difference and the performance between a clear or a white plate. And he clearly pointed out that if you have the option to go for a white plate will usually give you a much better performance. If you have a very stable and solid qPCR protocol and also expecting a large signal intensity, you can obviously also go with a clear plate. Just make sure that it has optical brilliance and high quality. So, and as you can see, these are like different parameters for the plate frame and the plate well, which cannot achieved with the same type of plastic. So this is why I am a big fan of a plate made of two components. For Eppendorf, these are the so-called twin tech plates. So they come with a polycarbonate frame, which is very rigid, which is very strong. So it can withstand heat, it's not warping, it's not shifting, but it comes with a polypropylene well, and this polypropylene well ensures perfect heat transfer and very fast PCR runs. Right. Then a lot of you might have seen very different PCR plates. Again, talking a little bit about nomenclature here, you can see 3 plates there from top to bottom on the top. So the first plate is a so-called non-skirted plate. Then we have a semi-skirted plate and a fully skirted plate. What plates you're going to use is usually dictated by your qPCR instrument or also by your PCR instrument, depending on what you're doing. Usually it's best if you have obviously an instrument which can take all of those plates. The difference between those plates is usually the volume that you can use with those plates. But there are some general things that you have to keep in mind. And I've listed this as grippable and stackable. So with grippable, I mean grippable by a robot arm, not by a human hand. Obviously, a human hand can work with all of those plates. So a non-skirted plate, the grippability is very limited by pipetting robots. And those plates are obviously not stackable. With stackable, I mean, if you put 2 of those plates on top of each other, can you build a stack without having any problems and risking your sample? Then we have a so-called semi-skirted plate. So this has a little bit of a bigger frame. This plate is perfectly grippable, but not really stackable. And then we have a so-called fully skirted plate. And those plates are both, they are grippable and stackable. And then sometimes you come across the term high profile and low profile plates. This basically means the geometry of the well, and it's actually pretty easy to remember when to use what. A high profile plate is usually used for high volumes, and the low profile plate is usually used for low volumes. So with this short introduction, I hope I can help you to choose the perfect qPCR plate for your assay. I know that lab reality dictates that there's usually a mix of consumables within a lab, and some consumables are of very high quality, and some consumables are of a little bit of a lower quality. So I can always encourage you to mix and match, make up your mind. What is the most important step of my workflow? How should the consumables perform for this step? And then I'm going to choose my highest quality consumable for the most important step. Obviously being consistent between experiments, so not swapping your plate for every experiment, because this can have influences on your results. And I also want to end with one tip for plate sealing, because I was always encountering some problems with plate sealing. So my tip is that if you want to store a plate, use foil, like this silverish aluminum looking foil for long-term storage, and use film, which is clear for detection if you run an experiment. Reason behind this is you want a clear film to get perfect qPCR results, but the foil is better suited for long-term storage because it is pierceable. You can just pierce through with a pipette, get to your samples. If you always want to remove your film, you will most likely end up with cross-contamination. Okay, and before I hand over to Rafal, as we at Eppendorf, we are constantly trying to improve our products. I have 2 questions for you which help us to make our products better. The first one is, are you doing your qPCR setup manually or automated? Are you doing your qPCR setup manually or automated? Manual, are you doing it manual but you want to automate it, or are you do your qPCR setup fully automated? Just quickly repeat the question. Are you doing your qPCR setup manually or automated? The answer options are manually, but I want to automate, or fully automated. And I will give couple of seconds, maybe 1 or 2 minutes for you to answer the questions. And we are very grateful that you participate here and that you give us your answer, because that really helps us to improve products and also develop new products. All right. We have a lot of answers coming in. And for those of you who are interested in the audience, most of the people are doing a manual qPCR, but among them, there is quite a number who are thinking about automation, which is obviously a very smart move, as also Professor Pfaffl pointed out. All right. Okay. So thank you for that. And then we have a quick second poll, and that leads me to something I've discussed already, would you consider the sealing of plates as an error-prone step in your workflow? So would you consider the sealing of plates as an error-prone step in your workflow? The answers are no, I hardly have difficulties with sealing. Yes, the sealing often causes trouble, or I do not seal my plates. So I will read the question one more time. Would you consider the sealing of plates as an error-prone step in your workflow? No, I hardly have difficulties with the sealing. Yes, the sealing often causes troubles, or I do not seal my plates. Again, thank you for every one of you out there answering those questions. It really helps us to get better. There's a few more answers coming in. Thank you for that. So it feels like there's a little bit of a 50-50 situation here between people thinking that sealing is an issue or is sometimes an issue, and those who are fine with their sealing technique. All right. Thank you very much with that. I hope I could give you a quick insight in consumables for qPCR, and with this I'm going to hand over to my lovely colleague Rafal for more topics.
All right, team. Thanks a lot. Glad to be here. Hope everyone is still awake. And it's indeed a lot of stuff, and it's a lot of important information we just received from Professor Pfaffl and Tim. I would like to dive in a little bit more into detail into the world of plastics, and I will show you probably some interesting and new data for you about how actually the very properties of the material, the quality of material may influence, of the plastics may influence the qPCR assays. I will talk mainly about leachables, probably topic already heard by of some of you, and unspecific sample loss. So let's go directly to the slides. As you see, this is already what Tim has talked about. Plastics have direct influence on the assays. And there is a lot of different aspects of plastics which might be taken into the consideration. So basically there's material, production process, quality control, purity grades, mechanical features, which we all know, so lead, centrifugation, stability, but also smart features, low binds. I will talk in my slides predominantly about the first, so the material, and the last, the smart features. And all of these parameters have dramatic impact on the sample, the safety, reliability of the experiments, and finally, cost and time. By the way, we will provide you basically this presentation in detail, so probably as PDF or also as a recording. So all of the details which we will not or did not talk about, you will still have a chance to have your own copy and go into the detail if you wish. So some important definitions at the beginning. Leachables are basically chemical substances which are in the material and released under specific experimental conditions from the consumable into the sample, reagent, or assay. And these conditions vary from experiment to experiment. So obviously you will have different amounts and different compositions of leachables in your reactions or samples dependent on the experiment. And examples here are, for example, leaching from microcentrifuge tube into stored DNA sample, which may increase during centrifugation because the tubes heat up or under physical stress, but also during the storage. Another example is leaching from PCR plates into qPCR reaction. The plates are being heated up and cooled down in many cycles, and as you will see, actually, I provide some examples on this. It may produce leachables in your reaction. And there is a similar term being often used. These are the extractables. They're used often in biotech and pharma areas, and these are the maximum amount of all of the substances which can be leached or extracted into the sample. So this is the worst case scenario. Typically in life science fields, they're not really relevant. So leachables got attention in the recent years from the scientific community. There have been a lot of different articles in the good journals in Nature, in Science, and basically the importance of leachables was largely underestimated by the researchers. So most of the scientists knew there is a certain impact of consumables on the experiments, but they actually underestimated the magnitude of these effects. And all of these papers basically emphasized growing importance of consumables in hampering the experimental results, in spoiling the experiments, and leading to change in perception, where high quality consumables are increasingly important in the experiments, particularly sensitive assays. And qPCR is one of those examples. So let's show some data. Here we have a nice article from Biotechniques published by Louis and his team. And what he did actually is very simple. He took two 0.2 milliliter PCR tubes and just water was cycled as a normal PCR reaction. So 30 seconds, 95, then 60 degrees, 30 seconds, and elongation 60 seconds. And astonishingly, what he have seen is when you put these water samples into biospectrometer, he saw some spectra. And these leachables strongly resemble nucleic acids. They have very nice peaks at, let's say, 260, the type 2 curve, and this type 1 is a little bit earlier. But both substances basically could be mistaken for being nucleic acids. We took this experiment and kind of wanted to quantify this effect. And this is what you see here in the first panel above is the absorption spectrum of water samples incubated in conical tubes just for 30 minutes. And you have different tubes from different manufacturers producing quite astonishingly high amounts of fake DNA. So basically this is not the DNA what you're seeing here or nucleic acids in general, but it's just the leachables which absorb in a very similar region than as nucleic acids. We quantified this fake DNA in the tubes, and basically here what you see in the panel below is the fake DNA concentrations in conical tubes taken from the curves above. And these amounts are pretty substantial. We can go up to 5 or even above 5 micrograms per milliliter. So of course, it will influence downstream applications. When you do PCR or when you purify nucleic acids and you see fake DNA, it will dramatically influence the downstream steps. We took it next level and we asked the question, how is leaching doing in the plates? Because plates, you have many wells at once, and basically there are two problems: the amount of the leachables per se and the variability or variance between the wells. And this is what you see here in panel A. We plotted here the average spectra obtained from 3 independent plates. So it's the average spectrum from 3 different plates. And you see, in general, high average levels of leachables. We incubated just for 30 minutes, 95 degrees, and this is already quite high amounts of leachables. What is, however, much more important is that the variance between the wells is extremely high. So below you see as a control, we always use our Eppendorf plate, which is very low in leaching. And here's an example of plate A from one manufacturer. And what is clear here is there's a high variance of leachable levels between individual wells. And this is something when you speak about all assays actually, which take place in plates. And also qPCR is something which you have, you would have a nightmares from, because one of the aspects is that qPCR and any assay in a plate format relies on the fact that you have different samples in different wells, and you rely on controls, you rely on standards in different plate wells, and also the measurement has to be reproducible and not influenced by any other factors. And what you see here, there is a dramatic influence on the position of the well dependent on the different plates. So basically, there is a huge factor increasing the variability in your assays in the plate format. There has been a lot of buzz about leachables, and there's just a little extract of different papers where we have the author of the publication, which consumables have been shown to be to have influence on different leachable types and affected assay. And basically, what we see here, there is a lot of ligand binding assays. There is the enzymatic assays, also cell culture graph or cell culture assays, also this simple photometric DNA or RNA quantification. What I want to stress here is actually there are two classes of leachables which are critical, and these are red or yellow. And in the next slide, I will I'm basically showing those three classes. So basically, and to nail it and to tell you the truth, all of these additives are being predominantly used during the production process of consumables, simply to alter the properties or to reduce the production costs, to make it faster, to make it cheaper. And these three classes, so the uncritical, have to be added during the production process or during the polymerization of the material. Otherwise, you will not have stable polymer. And these are the heavy metals, stabilizers, and antioxidants, also pigments in case of colorful consumables. The second class is potentially critical. It's the clarifiers, anesthetic agents, and surfactants. And the third, the most critical class, which have been shown in the literature to be responsible for most of the negative effects, is the slip agents, biocides, and plasticizers. And these two classes, actually, this is good news, can be largely avoided during the production process. So they don't have to be present in the tube or in the plate. And basically, the recommendation, the first one is in red, strictly be aware of this, of the topic of leachables, and try to purchase consumables which are free of the critical additives, so slip agents, biocides, and plasticizers, and ask for the respective certificate. And of course, all around it, it goes into the direction of the high quality, pure consumables. So the clean room conditions and high levels of automation have to be used for production of consumables. High quality raw materials, so not recycled, without extra additives, has to be used. And so my personal recommendation is whenever you can and it's necessary, choose clear or white consumables, and also ask the vendors, the manufacturers of the consumables for lot-specific certificate done by the external laboratories. And also consider always smart consumables, low retention and low bind, as we will see in the next slides. So just the take-home message here is topic of leachables. Be aware there are substances going out of the, I'll go back to this slide. Be aware that these substances may dramatically influence your experiments. qPCR is definitely one of those assays, and be aware of this and choose consumables which are certified free of these substances. Now, there is another topic I would like to talk about is unspecific sample loss. Not many people are aware of this. Let's start with the low retention. And basically, what you see here is a pretty common situation. You pipette your master mix for qPCR or PCR, and you always calculate a little bit excess, let's say 10, 20%. And this is due because detergent containing or viscous liquids, which is exactly the master mix, polymerase or PCR reagents, have tendency to remain in the tip. And this sample retention can sum up to 20-30% loss of your reagents in samples. But even worse than that, it might contribute to the huge pipetting errors. So the liquid retention in your tips are cumulative and basically can cause that your pipette and the tip will be severely out of specification. So will perform much below the specified parameters. And by this, compromise, of course, the precision and reproducibility of your assay. And to basically address this issue, we invented something which we call a low retention material at Eppendorf. It's ultra hydrophobic treatment, basically increases, as you see here in this molecular picture, let's say, or rendering, increases the surface tension and basically allows absolutely free retention-free pipetting of difficult liquids. And this is what you see here. Here's the standard tip. Here's the low retention tip. It basically completely went out of the tip. And so this may dramatically improve the performance of sensitive PCR and real-time PCR reactions, NGS library preparations, but also sensitive diagnostic procedures, including the hot topic coronavirus protocols. So remember, guys, sensitive assays always use low retention consumables, low retention tips. I will go perhaps to the video, which basically shows you nicely how does it work. If you just click in the middle, I hope it will work. On the play button, you will see how it works. I hope it will be possible. So just click in the middle. And let's see, on my side it's still working. Basically, you see on the left side, conventional tip, and there is a huge retention of the liquid. On the right side, there is low retention, and there is all of the liquid has been removed. And this is what happens at the molecular level. As you see on the right tip, this high, the low retention surface basically is not getting in contact with the liquid. All right. I hope you enjoyed the little video. So let's go to the next slide. We quantify these effects and pipetted simply water in different tips from different manufacturers and measured how much water is being remained in the tip, and these effects are pretty strong here. So we have different manufacturers. In some cases, it's going above up to 30 microliters. So these effects are pretty large. And I have another example here where we compare low retention tips to different tips and different volumes. And as you see, the blue bars is the larger tip, larger volume, and the magenta is lower. And these effects are much higher for the lower tips. And this has direct relevance. Here for the PCR, here we were dispensing PCR buffer, 1% SDS. And you see the bars are really high for the low volume tips, so 10 microliter tips, which is actually nowadays pretty often used pipetting the qPCR reaction, since the trend is to lower the volume of the reaction. And here, low retention tips, both volumes are very low volumes retaining in the tip. So this is basically, we were talking about the low retention. Last but not least, low binds, and this is something not most of the people are not really aware of, just in analytics perhaps, or in high-end laboratories. What is happening is the biological material, the biomolecules, are basically swimming happily in the aqueous solutions. They're covered by the water sheath or water layer, and outside they have hydrophilic and inside hydrophobic domains. And what happens when you put those little happy molecules into the standard polypropylene, which is highly hydrophobic, is basically they bind. So you have sample loss, but also denaturation and loss of activity. And this happens all the time in all of the life science experiments. So we address this issue with ultra hydrophilic polymer, where basically the molecules are prevented from binding to the plastics by the layer of water molecules. And this prevents the unspecific binding and also maximizes the sample recovery and activity of your samples. It's very important for low volume or low concentration of your samples, which in many cases, for example, in diagnostic samples is really important. And again, a little video to show you how actually this works on the molecular level. If you just click in the middle, this film will start. So we have standard polypropylene tubes. It refers both to protein and nucleic acids. So the tubes can bind both types of molecules. And we have two distinct materials addressing this, the nucleic acids and the protein samples. So here you have the protein samples being absorbed or to the wall of the tube. Here is the DNA, and here is the low bind material, which show at the molecular level what is happening. Again, DNA and proteins and PCR clean. So basically all of the low bind consumables are PCR clean. And this is what I mentioned, the water molecules prevent from binding the proteins into the surface, and the same for the DNA molecules. This increases dramatically the recovery rate and basically assures that activity of the biomolecules is maintained. All right, so this was the little film. And so I'll show some data. So here we start protein samples, fluorescein labeled BSA, in different tubes. And over the course of 96 hours, you see the concentration in Eppendorf low bind tubes is pretty stable. At the same time, in most of the tubes, within already the first 24 hours is really dropping down, and you have basically most of the sample is being absorbed to the tube. This effect is even more dramatic due to the surface to volume ratio in plates. And here we show the data from 384 well plates, where basically within first 2-4 hours, most of the samples in case here is the fluorescent labeled BSA are being unspecifically absorbed and lost to the experiments just because it is in the well made from standard polypropylene. So this unspecific absorption can lead to a dramatic decrease of the sample of the biomolecules which are available for assay in the downstream applications. And I think this is the last slide. So basically, as a wrap-up and recommendations from my side is choosing right consumables has dramatic impacts on the assay to perform. Consider that these factors may really contribute to increase the performance of your experiments. So low retention may increase the reproducibility and reduce the loss of expensive reagents. It might improve performance of PCR and real-time PCR applications. Low bind plastic will decrease the unspecific binding. So basically, you will have maximized sample recovery and activity. And finally, regarding the leachables, high-quality consumables free of critical additives may significantly reduce the risk and negative influence on the experiments. That will be it from my side. We're already 9 minutes over the time, but I think it was worth for you staying with us. And I give over to Tim and Professor Pfaffl for final word.
Hello, everybody. I just want to bring back slide number 16. If you're interested to download the iBook, and I think I have to do this with the, yeah, here we are. just click the link and you can download it for free, or you can also download on the web page a PDF version of the book if you don't have any Apple devices. So you have to log on to the MIQE site, and the MIQE site is beyond this nice cartoon here, MIQE Gene Quantification.info. There you can also find a link to a PDF. And now I hand over to the moderation for the discussion, I think. Thank you very much.
Yeah, thank you everyone for staying with us till the end. I know that we did run overtime a little here. I appreciate all of you staying with us. There were a lot of questions coming in through the webinar, and we are not able to answer all of them now live. So due to the advanced time, I will pick 3 questions, but be assured that we will answer all of your questions via email. So you will receive an email answering the questions that you've typed in during this webinar. Just give us a little time to answer to you. So the first questions that we have here will go out to Rafal. And it is, what is the difference between the Eppendorf protein low-bind tubes and the DNA low-bind? Can I use protein low-bind to do RNA-DNA extraction as well as all my qPCR? Or should it be better to use the DNA low-bind range?
Yes, this is the quite common question we always receive. And there is the difference between those 2 polymers. So they are specifically designed and optimized for nucleic acids and protein/peptides samples. So the DNA low bind will have effects for storage of DNA samples. So I would definitely use DNA low bind for storing your material, and also when you have very diluted samples. So for example, we very much recommend DNA low bind for forensic applications. In case of qPCR, I would say protein low bind might have better blend of properties, and what we have seen is that using protein low bind in PCR, qPCR sometimes performs even better than DNA low bind, and there is the reason for that. The protein low bind has to some extent DNA low bind properties, and of course the proteins remain intact. So since the qPCR and PCR is pretty complex reactions when you have both, I would probably, if I would recommend and choose one, I would stick to the protein low bind.
All right.
Is that?
Thank you for that, Rafal. The second question is directed to Professor Pfaffl. So what troubleshooting should we do if we have big spread results for only one gene in the plate? It is a PCR RT for gene expression.
If you only have one gene in the plate, which has a huge variation, all the other genes work perfectly well. It couldn't be due to the plastic, of course. It also tells us that the reagents are working because some genes have low spread, hence have nothing to do with the platform, with the plastics, with the consumables at all. The big spread for one gene could only be a problem of primer design, meaning that the primer did not fit perfectly, or there are mismatches in the primer sequences, and hence you got a big spread from for this particular gene.
Okay. Thank you for that. And I will personally answer the last question, and it is why is a white plate better than a clear plate in its performance? Is there any reason behind it, or is it just experimental observation? Yes, there is a reason behind it. It is not just experimental observation. The reason behind it is if you have a white plate with a white well. If you think about a qPCR cycle, usually the excitation with the laser as well as the readout is from the top. So basically, if you have a white well, 100% of the emitted light due to the excitation will be emitted in only one possible direction, which is top. So you will get the maximum signal recovery and signal strength, so to speak. And a second reason why white plates are performing better, this is in some cases, and it's not true for all qPCR machines, is that if you have a white plate, you don't have, let's say, a leakage of the fluorescent signal between two wells. So you only get the fluorescent signal from one well and not from the neighboring wells. I do hope this answers the question as well. I apologize that this time we can only keep it to 3 questions. There are a lot of other really brilliant questions we have here, but as I promised, we will answer all of your questions via email. With that, also, I will say goodbye. I thank you for your attendance, and I'm handing back to Chrissy.
Thank you. And thank you, doctors, for this valuable information you provided us today. It definitely prompted some interesting questions. Now, I do want to remind our audience, we had quite a few questions that we were unable to answer. Those questions will be answered. Don't fret. They will be answered, though, via the email address you provided at the time of registration. Now, I want to thank again Dr. Tim Schommartz, Dr. Pfaffl, Dr. Grzeskowiak. Gentlemen, do you have any final comments for our audience?
Let me start. I really appreciated it. It was very interesting as well for me to learn about the impact of consumables and also from the Eppendorf team. And yes, it was great, I think.
Also from my side, thanks a lot. And just to remember about consumables. And I basically send you warm greetings from Hamburg and stay healthy.
So then I go last. I can just also say thank you for attending this webinar. I hope all of you are safe and healthy. I wish you a great rest of the week. And if you have any questions regarding consumables or any other Eppendorf related products or workflows, do not hesitate to contact us anytime. You can find contact information on www.eppendorf.com. Thank you.
Thank you. A final huge thank you out to our speakers, Dr. Tim Schommartz, Dr. Michael Pfaffl, and Dr. Rafal Grzeskowiak. Thank you for your time today. We'd also like to thank LabRoots and our sponsor, Eppendorf, for underwriting today's educational webcast. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. In that email, you will also find a link where you can download your certificate for attendance if you would need. We encourage you to share that email with your colleagues as well who may have missed today's live event. That's all. Thank you for joining us. We hope you stay safe and healthy. Have a great day.